It's my great pleasure to introduce Dr. Rosa León Zayas, currently at the University at Willamette University as an assistant professor. Rosa actually started her academic career at the University of Puerto Rico, getting her Bachelor of Science degree in industrial microbiology back in 2007. And then she went from there to the other coast, to University of California at San Diego at the Scripps Institute of Oceanography, where she obtained her Master's of Science with Doug Bartlett in 2010, and then stayed in Doug's lab to get her PhD in 2014, working on uncovering the genomic properties of microbial life in the deepest portions of the Atlantic and the Pacific Ocean, one cell at a time. She then moved from there to do 2 postdocs at the University of Delaware on the Lewis campus I'm very familiar with. I was at Farm Point for a number of years and went over to Lewis quite regularly, a lovely place. The first postdoc advisor she had was Dr. Biddle, and she worked on several projects with Jen, everything from You Are What You Eat, the impacts of fish on coral reef microbiomes, to the metagenomics and metatranscriptomics analysis of the Tonga Trench microbiota. She then did a second postdoc, also at Lewis, with Dr. Dixon, discovering and characterizing the novel and abundant bioactive genomic signatures in environmental samples from Fiji coral reefs. He said she's since moved on from there to start her assistant professorship at Willamette University up in Oregon. And with that, I would like to turn it over to Rosa to talk about the environmental influences on microbial communities' structure and metabolic potential across the depth transect in the Tonga Trench. Thank you very much for doing this, Rosa.
Thank you, John. That was a very kind introduction. I'm very excited to tell you some more about the work that I started as a postdoc in Jen Biddle's lab, and that I've continued since I've started my assistant professor position at Willamette University. Before I begin, you will see this slide at the end again as an acknowledgement, but I just want to take a second to thank 2 important people. Doug Bartlett, like John mentioned, basically allowed me to go and be the lead scientist on this super exciting cruise to the Tonga Trench on my 5th year of grad school, so that was quite a leap of faith, and I really appreciate him taking allowing me to do that. It was a really incredible experience. And thank you to Jen Biddle and her lab for allowing me to come and work on this project and learn all the things about metagenomics over there. So with that, I'm going to shut my camera over here and tell you more about the Tonga Trench. So what you see here in this first slide is not necessarily the trip that we took to the Tonga Trench, but in a way it's somewhat similar. What you have here in the background is the Puerto Rico Trench, actually. And this instrument that I'm holding on to, partly so it doesn't fall off and partly so I don't fall off, is a lander. And there'll be a more detailed diagram or picture of this lander and what it's composed of. But this type of instrumentation allows us to sample into the trench either water samples or sediment samples. So, sorry about that. So why do we care about trenches? Trenches, or the deep waters in general, have been studied for the number of years, maybe a couple of decades now. And this is in the top a diagram of the metagenomic and metatranscriptomic analysis that have been done all the way up to 2004. The yellow dots represent metagenomes. And basically in the y-axis you have depth, and in the x-axis you have time. So the deepest metagenome that you see here is by Emily Elo, which was published in 2011. This metagenome was collected from the Puerto Rico Trench, and it's the deepest metagenomes that we have that's been published so far. We certainly learned a number of things from this metagenome, not only about community composition of a deep trench environment, but also about potential metabolic function. And if you look at the diagram of on the right side at the bottom, you can see there that the hadal zone, or what we call the trench environment, truly starts at about 6,000 meters. So this metagenome that Emily collected and published is barely the surface of the very beginning of the hadal zone. So there's a lot of depth in organisms and functions into the deep trenches that we truly don't understand very well yet. Most recently, John Tarn and colleagues published an article that described the microbial community composition using 16S amplicon sequencing, and they basically suggested that the microbial community within the trench was different from other more shallow environments, and within these two different sites in the trench, the Challenger Deep and the Serena Deep, the microbial community was rather different. It also is different based on size fractions. And that was quite interesting. Some of the most abundant and prevalent organisms in the trench that were published were some gamma proteobacteria, epsilon proteobacteria, alpha proteobacteria, and also some marine microbia. So those were some of the most abundant organisms that were found in the Mariana Trench. Within the Puerto Rico Trench, from Emily's study, we were able to see that the Puerto Rico Trench, when we look at the metabolism, is most similar to other deep environments that were published beforehand. So what you have here is the Puerto Rico Trench and how it's related functionally most closely to 4,000 meters in the ocean and 3,000 meters in the ocean in the Mediterranean, and more distantly related metabolically to the surface oceans. So that gives us a sense that the potential, not only the microorganisms may be different, but the functionality of these microorganisms seem also to be different in the deeper ocean. On the right is just a description of the microbial community composition, and interestingly, bacteria definitely dominate the community in the Puerto Rico Trench, about 90%. And archaea is made of about 3% of the community. Within the archaea community, Tom archaeota seem to be most abundant. And within the bacteria community, certainly the proteobacteria are most abundant, with alpha proteobacteria being the dominant member of the community. So we wanted to understand how the Tonga Trench may be different from this site in the Puerto Rico Trench, given that it's a completely different ocean, and not only that, but also the Tonga Trench is much deeper. So for that, we went on this expedition to the Tonga Trench. We were able to recover samples from the deepest, not from the deepest part of the trench. The deepest part of the trench is further south within the trench itself. And we collected samples from at a depth of about 9,100 meters, kind of further north in the trench from the deepest part. We collected 3 water samples using a C-DEBI rosette, 400 meters water sample. The idea with that was to try to get surface samples that was devoid of light, and then 2 mid-water samples at 3,000 and 5,000 meters. We then collected a deepest sample within the trench using a lander, which is the instrument that you saw earlier in the picture. And it's also, this is the actual lander that we were able to use. Landers are a really amazing technology that allow us to collect samples at full ocean depth. And what you see here is at the very top is a glass sphere that is full ocean depth pressure rated. Within this glass sphere, there's all the electronics that are necessary for collection and release of the weights. In the middle is a hard hat. Inside of the hard hat is also another pressure rated sphere that is mostly used for flotation. And in the middle of the instrument, then you have your sampling device. In this case, what we had was 2 30-liter Niskin water bottles. And these Niskin bottles go in open, and once the time control has run out, the electronic control releases a electric current that burns a burnt wire to which the water bottles caps were attached. And this releases the weights and caps the water bottles, and then this instrument comes floating back up. And then you just go fish it in the ocean. truly is a bit more complicated than that, but as a whole, it's a fairly simple process. What you have at the bottom is a pressure sensor, which allows us to understand exactly how deep this instrument went. So this is how we collect water samples in the trench. We also had the fortune of being able to recover a gravity core from this environment. This is probably one of the deepest, if not the deepest, gravity core that's ever been collected. It was to about 9,100 meters within this deepest part of the Tonga Trench, within the Tonga Trench, which is about 9,100 meters. And we were able to collect about 2 meters of sediment. This probably was the most beautiful scientific moment of my life. It was definitely quite a risky move to be able to collect this gravity core, and it was fairly successful. So this is me and another of our grad students in the Bartlett lab, Logan Peoples. So we collected 3 different samples for metagenomics from this about 2 meters of sediment. And what you have here is a simple sediment smear of this sediments that we collected. And I hope what you can notice is that the sample at the surface, or what we're going to call 0 meters below the seafloor, and the sample at 2 meters below the seafloor seem fairly similar. The grain is quite fine, so it looks like fine sand, while the 1 meter below the seafloor, it looks rather different. It's very mineral heavy, much more coarse grain. That was really interesting to us, and we certainly set out to try to understand why this might be different, and if that has any influence in the microwave community. We did some chemical composition analysis, and we have here total organic carbon, nitrate, ammonia, and phosphate, and some physical parameters like temperature, salinity, oxygen, chlorophyll. Also at the bottom, you have that description of the differentiation between the 3 different grain-like conditions of the sediments that we were able to analyze. The gray lines that go across denote the samples that the depth at which samples were collected for metagenomics. Some of the things I want to point out also is that at the top, the scale of carbon, nitrate, and is very different from the bottom, right? So you have the water samples at the top, and the scale itself is different from the sediment samples at the bottom. Something to note is that the total organic carbon certainly goes down as you go down into the water column, except for the 9,000 meters, which is rather high in comparison to all the other samples. This is likely due to the fact that when the weights of the instrument hit the bottom, there's always a large plume of sediment that gets kicked up into the water column. And it's likely that some of that material ended up in our water bottles. So I think that's the reason for that higher carbon composition. Nitrate certainly increases with depth, which is something that is expected. And another thing that I would like to point out is that in blue, you have the oxygen. And at 400 meters, inadvertently, we were able to actually collect that sample that is the lowest oxygen composition within the water column. So we happened upon a low oxygen zone, and those were the samples that we were able to analyze. The samples were analyzed using metagenomics, and for those of you that don't think as much as I do about metagenomics, I'm just going to describe this very briefly. We, as I mentioned, went onto the Tonga Trench and collected water and sediment samples, and we processed all this DNA and sent it out for sequencing, and ended up with small pieces of sequencing that belong to all organisms within this environment. With these small sequences, you can then do all kinds of interesting things that will give you really fascinating information. Among those things is studying the reeds themselves and try to assess the community composition and the abundance of organisms within the environment. You could also take those reeds and put them back together and make longer contiguous sequences, or we call them contigs, in which hopefully you'll be able to get a better sense of the functional potential of the organisms in the environment. You can also take those contigs and aided by the small reeds and try to extract from the community individual or at least population like organisms. So you can get draft genomes from the contigs that were generated and also utilizing the small reads. So you can look at the community composition of the whole metagenome, all the organisms there. You can try to understand better the metabolic potential of the community. You can also look at individual genomes within or organisms within the community. So first, I'm going to tell you about what we learned about the community composition in the Tonga Trench. Here is just a description of the domain level differences, relative abundance of bacteria and archaea in eukaryotes on the left-hand side, and on the right-hand side is a relatedness diagram using Bray-Curtis similarity at the phylum level. So if we look at the left-hand side, you can see that the viral composition and the eukaryotic composition is fairly stable throughout the water column and makes up less than 5% of the community. When it comes to bacteria and archaea seem to dominate that 400-meter low oxygen sample. And from there on, bacteria seem to increase with depth and dominates the community at about 90-some% once you get to the sediment samples. So the deep ocean seems to be dominated by bacteria, which is something that we also saw from Emily Elo's Tonga Puerto Rico Trench metagenome. When we look at the phylum level relatedness of the different samples within the Tonga Trench, we again see that the 400-meter community is fairly different, likely influenced by the Eroiarchaeota abundance in that sample. We can see that the mid-depth samples are 3,000 and 5,000 meters seems to be most similar to each other. And this is also likely this difference is likely to be driven by the abundance of Tomarchaeota within this population. The 9,000-meter sample clusters within with the 3,000 and 5,000 meter samples. And the water samples are certainly different from the sediment samples. I think it's really interesting to note that the 2-meter and 0-meter below the seafloor seem to be more similar to each other than that of the 1-meter below the seafloor, which in a way can be related back to that potential of the different visual representation that we saw from the sediment samples. So now we know that not only the sediments look different visually, but also the microbial community composition within the sediment is quite different. If we take a closer look at the microbial community composition by class, we can see some finer definition of what kinds of groups seem to be most prevalent or abundant in this environment. In the 400-meter, methanomicrobia seem to dominate the community, followed by delta bacteria. When you move into the water column, alpha and gamma bacteria certainly dominate the environment. Within the 0-meter below the seafloor, flavorbacteria seem to be quite abundant. And at 1 meter below the seafloor, distinctly from the other samples, bacilli-like organisms appear to be dominant, as well as actinobacteria-like organisms. So that middle sample in the sediment, 1 meter below the seafloor, again, fairly different from their neighbors. So now let's assess the community using assemblies, so putting those pieces back together. When we look at how good these assemblies were, it's just some statistics that allow us to understand how effective the assemblies were. We have contigs, so how many long pieces were created and how big those were. Statistics like the largest contig, and at the very bottom, you can see how many bins were recovered or individual draft genomes were recovered from these samples. What I would like really to point out is that when you look at the 1 meter and 2 meter below the seafloor, these samples did not necessarily assemble as well as all the other samples, to the point that the 2 meter below the seafloor sample only generated about 19 contigs that were larger than 1,000 base pairs and no contigs that were larger than 5,000 base pairs. That, of course, translated into not allowing us to recover any actual genomes, any organisms, draft genomes from this environment. So not only the samples seem to be particularly the 1 meter below the seafloor seems to be different from the other ones, but also did not assemble appropriately. And I should mention that the amount of sequences that went into this analysis were very similar from each other. So I wanted to try to understand better what was going on here and if we could see patterns just looking at the DNA itself. And what we have here is an analysis in which we plotted contigs based on k-mer composition. So in a way, this is kind of what we do when we're trying to bin samples or create draft genomes. And what you see here is that these contigs that are most closely to each other or are more similar in terms of the inherent DNA structure are going to cluster together in this plots. And I think you can note here that throughout the majority of the samples for 400, 3,000, 5,000, 9,000, and even the surface sample at 0 meters below the seafloor, you can see groupings, patterns emerge when you plot these samples together. This is definitely not the case for the 1 meter or the 2 meters below the seafloor. So these communities don't seem to have recognizable patterns, which started to lead us through the past that maybe this sequences, this sample themselves may be potentially devoid of active microbial communities. To look into this potential further, I performed an analysis in which I was trying to understand how related the samples are to each other. So I took reads from each of the different metagenomes and compared them or recruited them onto the assembled contigs within each metagenome. So for example, in the first row, I compare all the 400 meter reads to the 400 meter assembled metagenome, to the 3,000 meter assembled metagenome, to the 5,000 meter assembled metagenome, and so on and so forth. What you can see here, of course, is that the 400 meter reads are most similar or recruit more reads onto its own assembled metagenome, which is something that we expect. But it doesn't necessarily recruit a lot of different reads to 3,000, 5,000, and 9,000. This is expected, given that we know that the community composition in this different water samples are fairly different. When you look at the 3,000 and the 5,000 samples, they seem to recruit reads onto each other fairly well. So those 5,000 and 3,000 meter samples seem to be fairly related, rather similar to each other. Similarly, when you look at the 9,000 meter sample, and particularly the surface samples and how those two relate to each other. So the sediment sample seems to be very similar to itself, of course, and also similar to the 9,000 meters. This is not a pattern that you see particularly within the 1 meter below the seafloor at all. There doesn't seem to be necessarily a recognizable pattern of what is most similar. Not only that, but it seems that the most dissimilar samples and samples that recruit the least amounts of reads is the sediment sample above it and the sediment sample below it. When you look at the 2 meter below the seafloor, also very interestingly, it recruits many reads to itself, but it seems to recruit more reads to the sediment sample at the surface. So there seems to be a connection between the 2 meter below the seafloor and the 0 meter below the seafloor that does not exist at the 1 meter below the seafloor. So there seems to be certainly something very different about that one sample at the 1 meter below the seafloor. Not only visually, it looks very different, based on the composition of the community, also very different. And when you look at the structured data itself, it doesn't seem to be similar to anything else. We have done some preliminary analysis looking at the chemistry itself of the different samples that we collected throughout the sediment column using PXRF. And while this is kind of like a rough look at what the chemistry of the samples or the composition of the chemistry of these samples may tell us, what we see in a very preliminary analysis, the 1 meter below the sea floor sample seem to have a chemical composition that is more related to volcanic tephra-like samples. And when you look at things that are immediately above or immediately below the sample, the samples look very different. They seem more like serpentinite-like or derived environments. So there's certainly something very different. And this may be related to potentially physical changes in the sediment column event, or potentially slumping sediment events, maybe. So we definitely are excited to look more into that in the coming months. So this is how the community composition looks like once you assemble the metagenome. So now I would like to tell you a little bit about what we learned about the functional capacities of these different samples. So this is a analysis that, thanks to Ben Tolley and some of the scripts that he's generated to look at these different metabolic pathways and the enzymes that are important and necessary for the community to perform these different metabolic processes. And in general, you see that when we look at carbohydrate utilization and some respiratory-like processes, first of all, you should know that the 0 meters and the 1 meter velocity flora is kind of most often devoid of metabolic function, and this is not surprising given the fact that the assembly was so poor. In general, you can see that organisms are able, as a whole, utilize organic carbon, and they have the capacity to have respiratory potential. If we look at the composition, the potential for degradation of perhaps complex organic carbon like chitin, this seems to be a very popular metabolic processes that these organisms have, particularly at the surface of the sediments. Carbon fixation is something that these organisms may be able to do. There's certainly genes associated with things like rubisco, and also associated particularly with the 3-hydroxybutyrate hydroxypropionate cycle, which is associated with Tamar-Kyoto-like processes. There seems to be, as we talked about Tamar-Kyoto, there seems to be the potential for ammonia oxidation where Tamar-Kyoto are most abundant. So that is definitely not difficult to believe. This also seems to be the capacity to do sulfur metabolism by many organisms in these environments. Methane metabolisms like methanogenesis seems to be very popular at that 400 meter in the water column, which is the low oxygen sample. But this potential for creating methane from trimethylamine seems to be a process that is fairly abundant within this environment. And also organisms seem to be able to utilize small molecules like lactate formate and acetate, so on, to do metabolic processes. So as a whole, I think what I gather from a lot of this information is that truly the microbial functionality in these communities is not terribly distinct from the 3,000 meters to the 9,000 meters. So while some of the metabolisms are certainly different, there's a lot of shared metabolic potential within these communities. So the last thing that we wanted to be able to understand is, can we recover some organisms, some draft genomes from these environments? And we recover around 59 or so draft genomes from this different metagenomes. And this is just a quick snapshot of what some of those organisms are. The different colors represent the different samples. So 400, 3,000, 5,000, so on and so forth. The size of the bubble represents how complete those genomes are. So how much of the different draft genomes we're able to recover. And then you can see them plotted in an axis of GC content and genome size. I will tell you a little bit more about the genomes that were recovered from the deepest parts of the Tonga Trench. This is some of the most complete genomes that we recover. As you can see, the completeness is not stellar, but we do have some organisms that are over 50% completeness. So, and I will tell you a little bit more about what we have seen as part of the metabolic function, which is what we have at the bottom of the slide. We see, for example, the 9001 bin is that Tomarkia-like organisms, which is definitely we're looking forward to analyze a little bit more. Seems to have a metabolic potential that we would expect from an archaea. For example, this V-type HPase, certainly very potential for it to be there. And organisms seems to be able to have some, in a way, some respiratory potential. Again, this different bins are not complete, so the fact that things are not there doesn't mean that they certainly not exist. There's a capacity for them to do some degradation of organic carbon, like chitins, particularly the marine microbia seem to have some genes that are associated with degradation of some organic carbon. The majority of these organisms don't seem to have a lot of potential for carbon fixation, except for that one archaea, which has a number of enzymes associated with the hydroxybutyrate 3-hydroxypropionate sample. And also the archaea seem to have the majority of all the genes associated with ammonia oxidation. So we expect in a way that it looks like other tom archaea that are able to metabolize ammonia. And within that, it also seems to have the capacity for nitrite reduction. Organisms also seem to have a certain amount of sulfur metabolism within these different metagenomes in the deepest parts of the ocean or deepest parts of the Tonga Trench, and are also able to metabolize some small compounds like acetate and ethanol. So in general, this is just a quick snapshot of some of the most complete bins that we were able to recover from the deepest parts of the ocean. And in the interest of time, I am not going to tell you much more about any of the other bins that we are actively working on. This is an ongoing project and we're hoping to wrap it up hopefully in the next few months. Among other things that we are working on from the Tonga Trench is the analysis of a fungal genome that we stumble upon. This genome seemed interesting to us, this particular fungi seemed interesting to us because when we did our initial taxonomy or phylogeny analysis, seemed to be most similar to other things that were recovered from environments like hydrothermal vents. So we are certainly interested in putting that genome together and try to understand what maybe the fungal community is doing in the deep trenches. We also have just acquired and started to play in the lab with the Minion sequencer, and this is one of our first run was to analyze the 16S RNA amplicon sequencing of the Tonga Trench samples, and this is just a quick snapshot of the data that we recover in the ongoing process of using the Minion. We're actively going through the data right now, so I'm in pins and needles trying to see what if we can compare the 16S RNA amplicon sequencing to the metagenomic data. And I wanted to mention before I finish here that a lot of the data that I have been able to analyze in the past year has been with the help of a lot of my undergraduate students. So I'm very proud and very excited to be able to say that I'm working to, in a way, train the next generation of the biosphere scientists. The pictures that you see here are pictures of my undergraduate students from my research intensive methods course last semester, in which I basically provided each of my students a metagenome for them to be able to learn to analyze and generate data from them. And many of the students had metagenomes that were from this metagenomes that I just told you about in the Tonga Trench. And in the bottom picture, one of my students, Josh Sakai, is actually presenting the combined effort of the Tonga Trench just last week at the Zayas conference. So I'm definitely very proud of them and very excited to be able to teach the students how to use the technology and learn more about the deep biosphere. So with that, I just want to give some big take-home messages. In the Tonga Trench, it's potential that in particular in the sediment samples, there's some geological or physical events that could have shaped the microbial community. We see that microwave communities change throughout the water column, of course, and it's all very different in the sediments. And while the metabolic potential changes throughout, the differences from the mid-water and the deep water functional composition, it doesn't seem that dramatic. So there's certainly a lot more to know and learn from this environment. So much more work needs to be done to be able to understand what is so different about the microbial community composition and metabolic potential of these deep trenches. And again, with that, I just want to finish by acknowledging again the Biddle Lab and Jennifer Biddle for allowing me to do this work. Doug Bartlett and the crew of Roger Revell for allowing me to take the samples. My students that were most involved with this project, Marcia Smith and Katie Lewis, and also funding from the Census from Deep Life and C-DEBI, of course, for funding my postdoc. And thank you for listening. Now I'm gonna turn this on. Okay, that's me. And I think I can open the chat. Can I open this chat? There we go. Thank you for whoever wrote, because it was very helpful to find this clicking. So I will take any questions if anybody has any questions. Oh, I have one question actually. Thank you. Someone asked, which I'm sorry, I have an allergy today, so I apologize for the occasional blooding of the nose. Somebody asked, which P. Savalia, that it seemed surprising that the bacteria dominate over the archaea in deep waters, and also surprising that there seems to be some eukaryotes and no virus. Yeah, okay, so in general, I don't personally, and based on the things that we have known from things like John Tarrant's analysis and a number of other applicant analysis that we see, that has been reported from the deep trenches, it's not super surprising that there's more bacteria than archaea. That's something that we definitely expect. It is interesting though that, as you know, that there seems to be more eukaryotes around. And I think this is in a way why we are interested in looking at that fungal genome, because there seems to be definitely a large abundance of fungal like genes that we recover. So I think people have done a fair amount of analysis and studies on the potential of fungal organisms or fungal-like organisms in the deep ocean. So I think it's definitely interesting to think about it. We will continue to look at that. And I think when it comes to the viruses, we just were likely to not recover them. There's probably not that are not there, but these samples were recovered and filtered onto a 0.2 micron filter. So it's likely that the majority of the viruses kind of like fell through the filter. So that's why we likely do not see any filters there. Okay, another question from the Biddle Lab. Hello, Biddle Lab. So, okay, so somebody asked from the Biddle Lab if I think that the difference in heavy recruitment from the 2 meters to the 0 meters below the seafloor is in part due to the small amounts of assembly in the 2 meter below the seafloor. And I think the answer is, yeah, very probable. It's very probable that the reason why there's more recruitment to the surface sample is because there's more things to recruit to, certainly. But still, when you look at that differentiation, those are the two things, those two samples are the two samples that are going to be the most recruited towards. So it still gives us that sense that the 2 meter below the seafloor and the surface sediment sample are somewhat more related to each other than the 1 meter below the seafloor sample. Any other questions? So, okay, somebody asked if I thought that the, oh gosh, hi everybody. Okay, somebody asked if I think that The similarity between the 0 meter and the 2 meter below the seafloor, and how the 1 meter below the seafloor is so different, could be due to mass deposition due to earthquakes. And I guess while I don't have a lot of concrete physical data to be able to say that, I think in hindsight it would have been really great to be able to take good stratigraphy out of the core that we collected. Again, I think it was the first time that we collected a core like this, so it was certainly naivete for my part leading this expedition, but it would have been really great to be able to have very concrete pictures of how the sediment column looked like. But without that information, I think I would like to hypothesize that there was some sort of physical event that for some potentially slumped a bunch of sediment onto that 1 meter below the seafloor sample. This can happen in a number of different ways. It could be due to earthquakes. It's also in a trench, so it is possible that sediment just slumps every so often. So I would like to think that some physical event happened that just kind of changed the community composition as a whole in that sample. Somebody asked me why I don't have red hair anymore. I wondered the same thing. I was also surprised. There was a question about the no evidence for fermentation. And I have to say that I was also surprised that there was not a lot of evidence for fermentation, particularly in the sediment samples. But this is just certainly a quick look at some of the genomes that we recovered. And a lot of the genomes are not super complete. So I don't think necessarily I could say that fermentation is not found. And Ben Tully asked, when is this genome going to be public? And my answer to you is hopefully as soon as possible. We're working really hard right now to put a little bow on this and study and submit it for publication. This will be ideal. Anybody else? Okay.
Well, there aren't any other questions. I'd just like to thank Rosa again for all of her time and effort and that she put into this talk, this wonderful talk. And thank you very much.
Thank you, guys. Thank you for listening, and I'm sure we'll see you soon. Bye.